Okay, so my talk today is all about the overarching question of my talk is remembering what happened to us and in what order, what happened when. So at the end of the day, we can easily recollect all the events, the major events that took place during that day, and we can put them in the right order. This thing happened first, about an hour later, I did this, and so on and so on. How does the brain do that? Well, we know that the hippocampus is necessary for us to perform that basic function. We know that because, for example, if you're asked to walk around a campus and asked to do a bunch of tasks like pick up a coin, drink some water from a fountain, buy an item from a cafeteria, and so on, once you return back to the base and you're asked what happened, you can easily list the tasks that you took, but also you can put them in the correct order. Shown here in black is healthy individuals. I can nicely correlate the actual order of things, the actual order of events with their recollection. If you have a hippocampal damage and you do the exact same thing, not only you have a problem remembering what you did once you return back to the base, but you also have a really hard time putting the tasks that you participated in the correct order. So shown here in white, it's completely uncorrelated what you report as first, second, third with what is reality. So we know that the hippocampus is necessary for creating a model of our experiences and the time it took between them and putting them in the right order. So the question is, how does the brain, how does the hippocampus create a model of that? How does it represent the various events that are taking place and keeping track of the time interval in between them so that you can stack them in the right order? Let's simplify the question a little bit. So when you're talking about a mouse or an animal, an experimental animal, you might say, okay, I have a sequence of sensory stimuli, like a very basic odor. It was followed by, let's say, a visual stimulus, then maybe another odor came on and so on, with some time intervals in between. And the question is, how does the hippocampus create a representation of all of that so that then it can be stored as memory and be retrieved later? Well, we know that the hippocampus generates representations that come more or less in two flavors, let's say. One is sensory representations, representations of the external world, sensory cues that are coming into the brain. So for example, if a mouse needs to pay attention to a sound that is followed by an odor, then the hippocampus generates a sequence of neurons that are firing one after the other during the sound presentation, taken over by a different sequence that fires during the odor presentation, and so on. So we have cells or sequences that cover these sensory cues, direct sensory inputs coming in. For the time intervals, we also know that the hippocampus generates, again, representations, this time internal, we call them internal representations. And those can be representations of place, the very famous place cells, or representations of time. We have time cells as well. For example, if a rat needs to remember a cue while it's trapped in a part of the track here for a few seconds, the hippocampus generates sequences of neurons that fire one after the other, like the one shown here. Essentially, for us, the decoder, keeping track of time elapsed into this entrapment here. So basically, these neurons, one after the other, tile the interval in between the events. So put these sort of two kinds of representations together, and you can say, I have the hippocampus generates these sequences encoding the actual sensory input. So let's call these sensory cells. And for the intervals in between, it generates sequences of time cells. So that if later on if I knew exactly what each cell here represents, I could just look at this sequence and know that, oh, there was an odor that came on here, and then 1, 2, 3, there was 5 seconds here, and then a visual cue came on, and so on and so on. So I created a model representation of the experiences and the temporal intervals in between. Now, people usually study either these sensory representations or time cells or place cells separately. So my question was, putting these things together, does the hippocampus generate these external and internal presentations using the same dynamics, or does it use different modalities for the two kinds of representations? So let's break this question down a little bit. So first, can sequences truly combine these representations of external cues and time intervals? To answer that, we needed a task that combines both. So we trained mice to perform an olfactory delayed non-match to sample task. The trial is initiated by an odor. It can be either odor A or odor B. And then 5 seconds later, again, an odor comes on. It can be A or B. The mice are trained to remember the first odor throughout these 5 seconds and compare it with the second one. If the two orders did not match, so it was an A-B or a B-A trial, they lick to get a reward from a lick port. If the two orders were the same, an A-A or a B-B trial, they reject, they don't lick because there's no reward. So in this case, we have an important sensory cue that the mouse needs to remember, and then an important temporal interval during which working memory is activated in order for the cue to be compared with the second cue. Okay, so if we want to, so then I talked about if the dynamics are the same for these two representations. So the question specifically is how stable are these representations across days, or if we change something in the trial and the context of this trial and so on. So if you want to talk about stability, you want to be able to track the neurons across days. So our mice are head fixed on a styrofoam ball, free to run whenever they want. They're under a 2-photon microscope. So I've removed the cortex overlaying the hippocampus, and I put a cover slip so I can perform 2-photon calcium imaging on the dorsal CA1 pyramidal layer. So if I do a nice surgery, I get thousands of cells here. In red, you can see interneurons. These are mice that express TD tomato in all their interneurons. So I can image the interneurons in the red PMT channel and then remove them from my analysis. Everything I'm going to show you is only from pyramidal cells, which are shown here in red. So as the mice are performing the task, we are recording nice videos like this and tracking the activity of these neurons. And finally, the last question is, so if these sequences really exist, do they emerge during learning of this temporal context and so on, or are they pre-existent sequences that are just getting assigned to these trials? And so to answer that, you don't need a task that will take months and months and months for mice to learn, because it's really impossible to track these neurons nicely for that long time. So luckily for us, the mice can really learn this task and perform really well within 5 days or so. Okay, so what do we see? So let's split the trials in the ones that start with order A, shown here in yellow, versus the ones that start with order B in green. So what we find is we have a sequence of neurons that are tiling starting from the presentation of the order and tiling the entire delay. And this sequence more or less repeats itself if the second order is the same as the first one. So you have a bunch of cells that are representing the actual delivery of the order. And these are followed by a sequence of neurons, the so-called time cells, that are tiling the entire delay. And notice that these neurons are quiet when the trial was initiated by the opposite order. So if you just focus on a neuron that's sitting there, these are average firing rates. Now, if you look at a neuron that's here, basically you look at all the trials, and here I've stacked the trials based on the sequence of orders. So this neuron likes to fire when order A is presented, and it doesn't really care if it's the first order in the trial or the second order. It fires right at the presentation of order A. So a neuron down here, on the other hand, fires about 2 seconds into the delay, but only after order A was presented, not in trials that were initiated with order B, where the cell is more or less quiet. For the case of the order B trials, you have a different sequence, another group of neurons that are doing the exact same thing, but just for the opposite order. So an order cell up there will be firing only when order B is presented in green, and a late time cell here will be firing towards the end of the delay, but only after order B was presented, not after order A. Okay, so that kind of answers the first question. Can sequences combine representations of external cues and time intervals? Yes, I've shown you 2 different cue-specific sequences that combine representation of both the stimulus and the time interval. Oh, sorry. So the second question is, how stable are they across days or changes to a temporal context? Let's start with the second part, the temporal context. So the easiest way to change the context is to just extend the delay, right? From 5 seconds, we just extend it to 10 seconds. Mice keep performing the task, but they are just suddenly introduced to a 10-second delay and they keep going. What happens to the cells? So this is an example of an order cell. These are all the trials of 5-second delay, and this is the firing rate of the cell during those trials initiated by the order that the cell encodes. This is an early time cell firing just at the onset of the delay, and this is a late time cell firing towards the end of the delay. Now, what happens when we suddenly introduce the 10-second trials instead of 5 seconds? The order cell keeps firing here. The firing rate drops a little bit, but it retains a significant field very nicely. The early time cell goes completely quiet. Late time cell becomes completely disorganized here and loses its field. So if you look at the sequence as a whole, you have a nice sequence for the 5-second trials, the exact same neurons stacked the exact same way for the 10-second trials. the time sequence here is gone, even though the odor cells are nicely stable. Okay, so you have a remapping of this sequence. I should say here that these trials are tiled by their own sequence. It's just a new set of neurons. The exact same thing happens across days. So we track the same field of view for multiple days, and we follow the neurons for the same neurons across days. And what do we see? Take any day, day X, and you find a nice sequence of cells. And if you look at the same neurons the next day or the day after, again, we stack here the neurons the exact same way. The firing rates here are pretty much conserved. So the odor cells remain odor cells with a nice significant field firing during the preferred odor, whereas the nice sequence of time cells is already jumbled up the next day and pretty much gone 2 days later. Again, each day has its own sequence that looks pretty much like this. It's just composed of new time cells, even though the odor cells are more or less the same. So stable odor representations across days or across changes, but flexible temporal representations. Finally, how do these sequences emerge during learning, if they do? So again, this is the performance of the mice as they're learning the task, and we image them as they're learning. This is the sequence I get from a bunch of mice on first day of learning. So you have some odor cells, and here's some time cells. I separated them by a dashed line. And this is the exact same mice imaged, the same field of view for each one, imaged 6 days later when the mice are now experts in this task, and you get about, again, some more or less 150 odor cells, but way more time cells. In fact, if you just count how many odor cells you have every day from these mice, you have a pretty much flat line here. Whereas if you count how many time cells you have, it nicely correlates with, it nicely increases across days. So basically the number of time cells is very nicely correlated with performance, whereas the number of odor cells is not at all. It remains fixed. You might say that this has to do nothing with learning, and it's all because we trigger these odor cells over and over and over again because we present these odors for many days. It has nothing to do with learning. So to address that, we repeated the experiment with completely untrained mice that received the exact same trials, but they didn't know what to do with these trials. They were not trained to lick for a reward and so on. On day 1 and day 6 from these mice, we get the exact same, more or less the same sequences. Same number of odor cells, number of time cells is completely flat. It does not, the number of time cells does not increase. So you need the active learning of the trial and the context to increase your time cells. So to conclude, time representations emerge or expand during learning of a temporal context. And just to put this all together, I asked, are external and internal representations governed by the same dynamics? And I hope I've convinced you that the answer is no. We get very stable representations, stable odor cells for fixed external cues, the odor coming in. Whereas for the variable temporal intervals in between these experiences, we have very flexible and dynamic and learning dependent representations. And I would argue that this combination of stability for stable things coming in from the external world and flexibility for variable temporal interval representation might be necessary for the hippocampus to be able to construct these maps of what happened when and turn them into memory. I would like to thank everybody in the lab that has helped more to some degree or other with this work, and of course Payman Golshani for his mentorship. This work was published in Neuron a few weeks ago, and thank you very much for your attention. I'm happy to take questions.
All right, thank you so much. We have maybe 1 minute for questions, and so while questions are coming in, oh, we have one already. Awesome. All right, so Kara, sorry if I'm mispronouncing that, Wiseman asks, I'm wondering why the time cells increased with training when the time between stimulus and requested response wasn't something that the mice had to learn, or did I misunderstand the task? The training was to respond based on differences between the stimulus, not to count time?
Exactly. That's a very fair point. So basically, what I claim here is that the time cells are not really keeping track of time. The hippocampus is not required to count first second, second second, third second, fourth second, and so on. What it needs to do is just remember the first order, right, throughout the delay. So what these time cells do is they sort of hold this information because they're order specific, the sequences, they hold that information up until the second order comes on. And what's happening here is that as the mice are learning this task, they learn that the first order is important and I need to remember it, and they engage their working memory more and more. The hippocampus seems to be throwing more time cells into this task, building these sequences so that this information is carried over and up to the second order more and more efficiently. That's what I think is happening.
All right, thank you so much.